In this video, we will discuss about development of male gametophyte. This is the sixth video of sexual reproduction in flowering plant. Now, to know the development of male gametophyte, we must know the structure of pollen grains or microspores. Microspores are the spores which are produced after meiosis from the spore mother cell, which we have already discussed in the previous video, and link will be shared in the description box for all the video of sexual reproduction in flowering plant. Then what is pollination? How does pollen grains are liberated? And how does these pollen grains they germinate to form male gametophyte? We'll discuss step by step. Now first we'll discuss briefly a structure of microspore or pollen grain, and we have studied its structure in detail in the previous video. But just to understand the development of male gametophyte, we'll again see what is the structure of pollen grains or microspores. Now, if we see pollen grains or microspores, they are haploid in genetic constitution because they are being produced after the meiotic process. Now, we can see here in the previous video we discussed about anatomy of anther, and we saw this is the sporogenous tissue, and this sporogenous tissue is diploid in nature. This will undergo meiotic cell division to form microspore mother cell, and microspore mother cell will undergo meiosis to form the 4 microspores or pollen grains. So each microspore is haploid in genetic constitution. Now this is bounded on outer side by a thick wall. This is called as exine. What is the exact chemical composition of exine and intine? This we have already discussed in the previous video. And this is the internal wall. This is intine. This is quite thick, and this is pectocellulosic in nature. This area is cytoplasm, and this is its nucleus. Now, this cytoplasm is rich in reserve food materials. Exine is quite thick, but it may be thin at certain places, right? Here it is thin, so this thin area is called as germ pore. This is the region from where pollen grain will germinate to form the pollen tube. Now this may be rounded like this, or this may be present in the form of a furrow. When this is present in the form of a furrow, then it is called as germinal furrow. Number of the this thin area also varies. Now here we can see 1, 2 and 3. Here there are 2. Now sometime it may be only 1. So a number of the thin areas or germ pores varies from pollen grain to pollen grain, from family to family, and that is also called as NPC system, number position of the apertures or thin areas present in the pollen grain. Now we have seen there are this is the nucleus of the microspore or the pollen grain. Now this nucleus will undergo mitotic cell division. Now this will undergo mitotic cell division. First this was being produced from the sporogenous tissue after meiosis, but now this nucleus will undergo mitotic cell division and 2 nuclei will be produced, right? And this is one nucleus which is called as generative cell, and this is also called as tube cell or vegetative nucleus or tube or this whole cell is called as tube or vegetative cell, and this is called as generative cell. And this tube or vegetative cell, this is actually rich in food materials, like this may be having RNA and proteins. But this generative cell, this is rich in DNA because this is going to form the male gametes, and this is poor in reserve food material, and these are tiny vacuoles present in the cytoplasm of the pollen grain. Now these microspore or pollen grains, basically they develop inside these anthers, right? Like we discussed the anatomy of anther, and we have seen this sporogenous tissue which lead to the formation of the microspores or pollen grains. Now this anther will break up or dehisce open. This is called as the dehiscence of anthers. Like we have discussed in the previous video of dehiscence of anthers, how pollen grains are liberated. Now these are the pollen grains which are being released from the anther after its dehiscence. So this release of pollen grains from the anther, this is also called as pollination. Now while they are present inside the sporogenous tissue or inside the anther, these pollen grains they start developing and this is called as pre-pollination development. What is pollination? It is the liberation of the pollen grains from the anther, right? And when their pollen grain are present inside the anther only and they are developing, this is called as pre-pollination development. And when they are liberated, then it is called as post-pollination development. So these are two terms: pre mean before liberation and post mean after liberation, right? So these pollen grains, when they are liberated, they will be dispersed from one place to another place. So there are different methods for dispersal. I will not go into the detail, but it can be with the help of insects, it can be with the help of wind. But ultimately, pollen grain has to reach to the stigma of the flower, right? Now, if we see the structure of gynoecium, right, this is ovary, this is style, and this is the stigma. Now, stigma will attract the pollen grains toward itself and they will fall on the stigma, and then these pollen grain, they will start germinating, right? So, pollen grain, they will reach the stigma part of the flower, or we can say female part of the flower. Now, next we'll discuss in detail development of male gametophyte. Like we discussed, there is pre-pollination development before the liberation of the pollen grain, and after the liberation of the pollen grain, this is called as post-pollination development. First, we'll discuss about pre-pollination development, when this is occurring inside the anther part only. Now, we discussed that there is present 4 groups of sporogenous tissue in the anther when it is dithecous, and when it is monothecous, there are 2 groups of sporogenous tissue. Now, this sporogenous tissue will get differentiated to form microspores or pollen grains, and ultimately all the layers, they get degenerated and we are only left with the pollen grains inside the anthers. Now, this area of the anther is thin. This is called as stomium. From here, pollen grains will be liberated. We can see here, here anther is breaking up to liberate the pollen grain, and this area is stomium or thin area from where these pollen grains, they are liberated. Detail we have already discussed. Now, when these pollen grains are being liberated, they are already developing inside the anther, right? So, this is called as pre-pollination development. Now, this nucleus, as we discussed, this will undergo mitotic cell division to form 2 nuclei. This is called as tube nucleus or vegetative cell, and this is called as generative cell, which is going to form the male gametes. And this is 2 cell stage having tube or vegetative cell and generative cell, right? So, this is called as 2 cell stage. Now, when pollen grains are liberated at 2 cell stage, right, or they may be liberated at 3 cell stage, this may develop into male gametes inside the anther only. When the development of the male gamete is occurring inside the anther, right, this is also called as endosporous development. So, pollen grains, they may be liberated at 2 cell stage, they may be liberated at 3 cell stage. So, this varies from families to family, right? So, this development of the pollen grain, when it is occurring inside the anther, this is called as pre-pollination development. Now, when these pollen grains, they have reached the stigma of the female part of the flower, then it is called as post-pollination development. Now, when they fall on the stigma, right, then they start further developing, like they were liberated at 2 cell stage, that is tube nucleus and then generative cell. Now, what will happen? This is the tube nucleus and this is the generative cell. Certain changes will take place. We can see here, there is deposition of the callus around this generative cell. Now, callus, we have already discussed what is callus. Callus is a polymerized product of beta 1,3 glucan, glucose unit. They are linked with the help of 1,3 glucan linkage, and callus is dissolved with the help of enzyme callase, right? So, here this generative cell gets separated from the intine wall of the pollen grain by the deposition of the callus wall. Now, later on, this callus will get degenerated and this generative cell will become free in the cytoplasm. Now, here there is no callus, and there are certain changes in the tube cell or in the vegetative nucleus. This has become like this, convoluted. This is called as convoluted. Okay. And this area of the cytoplasm is rich in reserve food materials, and these are the vacuoles which have developed inside it. Now, at 2 cell stage or at 3 cell stage, now this will fall on the stigma of the flower, right? We can see here, and this is the stigma. This is ovary, this part is ovary, this is ovary, and this is the style, and this is the stigma. And here pollen grains there fall and they start germinating to form the pollen tube. Now what changes occur when pollen tube is being produced, right? We can see here this is the vacuolated cytoplasm. I mean, this area is rich in vacuoles, so this is called as vacuolated cytoplasm. And this is formation of callose plugs. Callose, it will block block the area, and no development will take place in this area. This will be hindered. And here these are two male gametes which are being produced from this division of the generative cell. Now this generative cell has divided into two male gametes, right? So these are two male gametes here, and they are surrounded by a cytoplasmic sheath around it. And you can see here this is the green color cytoplasmic sheath present around the male gametes. So these are two male gametes, very important. And this part is tube nucleus, which has reached to the tip of the pollen tube. And what happens, how this pollen tube is being produced, we'll discuss in the next slide. Now these are two male gametes, then this is the tube nucleus. This area is rich in cytoplasm, and this area is plugged with the help of callose formation, and this will become non-functional. And these are the vacuolated vacuole present. This is the vacuolated part of the pollen tube, and this area will become empty. What happens actually, we'll discuss now, just to make you explain. See, this is the stigma, and pollen grains, they will fall over here, right? Now each pollen grain will start developing pollen tube. Now pollen grain will form pollen tube from the thin area, that is from the germ pores, and this will absorb water here and absorb secretions from the stigmatic surface, right? And when this will absorb, cytoplasm of the pollen grain will swell up, right? And from the thin area this will start forming like this pollen tube, like this. And this will further grow up in the styler tissue. And while this is growing in the styler tissue, this will absorb nourishment from the styler tissue. And this is this movement of the pollen tube, like this here pollen grain germinate, and this is pollen tube, this is moving, right? This is also called as chemotactic movement. Chemotactic movement. This is being guided with the help of chemicals, right? So whatever stigmatic secretion or stigma is wet or certain secretions are there, pollen grain will germinate, pollen tube will be produced. Pollen tube is always produced from the thin area, that is from the germ pores. And then pollen tube, inside the pollen tube we are having two male gametes. Now these are two male gametes. We can see here two male gametes, like in the previous we saw here. These are two male gametes, right? Now these male gametes will reach toward the female part, and these are the ovules. Ovules are having female gametophyte, and this pollen tube carrying the male gamete, this will reach toward the female gamete. Ultimately fertilization can take place, and that we'll discuss in the next videos. So pollen tube, if we see the properties of pollen tube, it is glandular, secretory and absorptive. While it is moving inside the stigmatic tissue, this will absorb nourishment from the styler tissue, and this will keep its path in a particular direction, and this also carries the male gamete, right? So this is all about development of male gametes. So we have seen here there is formation of two male gametes inside the pollen tube. So this is called as development of the male gametophyte or development of the male gametes. Now thank you for watching my video. If you like my video, please like, share and subscribe. And if you have any questions or doubt, please ask me in the comment box. Thank you.